So good morning, ladies and gentlemen. So it's my pleasure to be the last before the lunch break here. So I hope you're not too hungry here. Before I start with my talk, I would like to disclose that I'm a consultant of BRACCO and that I have joint patent applications in a strong collaboration with Fuji VisualSonics here. Peter Burns already gave a great overview on the ultrasound field and recent developments, and I think I can really catch up on his talk and go into some of the aspects a little bit more deeply that are somehow related to the field of theranostics. When I was asked to talk about theranostics and ultrasound, I was asking myself, what should I talk about and what is theranostics? Yeah? And if you look at the definition, so you see that there's an evolution in the definition of theranostics. So several years ago, when personalized medicine was one of the fashion words in science, theranostics came up and was defined as a science where you use a drug molecule that has diagnostic and therapeutic properties. Over the years, then personalized medicine became unattractive, so people invented the term precision medicine. Yeah, that also stayed for some time until this also became unattractive. So they thought maybe we can pick up theranostics and give that also the same flavor as the two other words. So the second definition of theranostics was then a form of diagnostic testing employed for selecting a targeted therapy. So it was still a direct relation between the drug and the diagnostic procedure. And then the most recent definition of theranostics was a combination between diagnostics and therapy, and that's what we all do every day with all imaging devices. So as you see, I can talk about everything here. But I will try to give you examples for each of the three definitions for the ultrasound field. Well, first we should think about what are the demands if we really want to use diagnostic and theranostic biomarkers that should help us in stratifying therapies. And I think the most important ones are these three. The information that we get should be quantitative, the information should be robust and reproducible, and it should have a direct impact on the therapeutic procedure. And if we now consider ultrasound parameters, we have quite a large variability of parameters that we can receive from ultrasound. So we can characterize tissue morphology. We've already seen some nice examples about that. So we can look for the contours of a lesion. We can look at grayscale. We can look at speckle. We can even use the artifacts of the images to improve our diagnosis. Then we have some functional methods like elastography, which is increasing in the field of liver diagnosis that can be added. And we have all the information that comes from the vascularization that we can receive either by using Doppler methods or contrast-enhanced ultrasound tools. Well, so we can say that ultrasound is really a very versatile tool and can be nicely applied in preclinical research and the clinics. And in addition to these multitude of parameters, it's also cheap and real-time. And it can be quantitative. Yeah? Even it's mostly quantitative, but it still is user-dependent and it has a limited reproducibility. And this is the reason why you find so few papers about radiomics and theranostics in the ultrasound field. So what can we do about it? So first of all, we have to work on making the lesion detection and lesion delineation more user independent. And this is just an example from work of a master student in my lab who uses artificial intelligence to improve the detection and automated segmentation of tumors. And you see here an example of a subcutaneous tumor lesion. And you see that the margin of the tumor is incomplete. So here, there's no pointer. Was there a pointer somewhere? No pointer, okay. But you can see that easily that on the bottom left here, there's a discontinuity in the discontinuity in the borders of the tumor. You also see that there are also some bright structures inside, which makes it difficult for an artificial intelligence system to really detect the borders. But now we've wrote an algorithm that firstly investigates what is really border and what is not border. And the algorithm starts to automatically remove everything from the image that is not part of the border. And then he starts slowly to analyze, you will see that in a minute, the border structures, the symmetry of it, and then it finds that there is some kind of discontinuity and it goes on with reconstructing it, and so you can automatically then segment the lesion. This does not work yet in real time, but we're working on that. It's only a question of computer power until this will work reliably. And if you now compare here the accuracy in lesion detection between the automated system and the patient. Oh, this is really kind from you. Thank you very much. That helps a lot. Here you see that we are very close to the user data, but with a much lower variability between observers. There are some outliers here where we have to look at, but this is certainly a problem that can be solved. So we can really take aid of computer power and artificial intelligence to improve reproducibility of ultrasound imaging. And if you have segmented such a lesion, we then go on with characterizing the lesion. So we want to get as many parameters out of it as possible. In this context, really the microbubbles are of great help because they give us terrific information about the vascular system in these tumors. And in this talk, I will show you how these microbubbles can be used to assess microvascularization, how they can be used for molecular imaging. In the final part, I will also talk about therapeutic applications. Here's just an example from a paper that we wrote several years ago here, where we used a multispecific tyrosine kinase inhibitor to treat squamous cell carcinoma xenografts. And we imaged these tumors with a high-frequency Doppler method without contrast agents and with contrast agents. And as you see, the images look quite different. You see in the pure Doppler method, you see just at the border, you see some bigger vascular networks, some of them infiltrating the tumors. But in the contrast scan, you see a more or less homogeneously vascularized tumor. And if you look by histology, what that means, you will find here that here you have the majority of immature vessels displayed with very slow velocities, while here you only see the larger vessels, which are stabilized with smooth muscle cells, which are more mature. So these mature vessels are not the typical sign of malignant tissue. You find them normally more pronounced in normal tissues. Yeah? And what happens now if we start to treat this tumor? You see the immature tumor vessels diminish from day 0 to day 3 and then further to day 6, while you only have a moderate decrease in the mature vessel fraction depicted by Doppler and almost no change then from day 3 to day 6. Now let's look what that means if we really track therapy responses of these tumors. So we can here clearly see that the therapy works, so the untreated tumors constantly increase their volumes, the treated tumors respond and decrease their volumes. Then we see that on both ways of ultrasound scanning, contrast enhanced and non-contrast enhanced, we see that the vascularization of the tumors slowly drop, almost in the same way. This is due to the fact that if tumors grow, the interstitial pressure goes up, then the vessels become hypovascular. So there's often, by the way, a high discrepancy between pathological microvessel density and the real vascularization that you find by ultrasound. And this is just due to that fact that about 30 to 60% of the vessels in a tumor usually do not carry blood. This is something that you should know. Yeah? So that's the reason why vascularization goes down. That's also the reason why micronecrosis develop. Then you see if you treat the tumors, you see a drop in vascularization in both scans, but then something strange happens. While on the contrast enhanced scan, you see a constant drop in vascularization over the whole observation period. You see that for the Doppler scan, vascularization between 3 and 6 goes up again and then is higher than in the control tumors. So what happens here? Is this an artifact? What do you think? Yeah? Or do we see a biological phenomenon that is potentially important for therapy? The latter is the case. Because what happens is, imagine I destroy the immature vessels and they are just gone. As a consequence of that, the tumor cells will die. So the tumor will start to shrink. But the mature vessels, they are not that responsive to antigenic therapies, so they may persist. And if the tumor cells are gone, these mature vessels will just draw closer until you will end up in a tumor with a higher vessel density concerning the mature vessels. And this is something that you can call vessel maturation. It's a little bit different than the maturation that Rakesh Jain described. Rakesh Jain said that if you apply antigenic therapies, the vessel will really turn the molecular profiles and get maturated. Here you have a passive way of maturation by just killing the immature vessels and then having the more mature vessels drawing closer. That's what you can nicely see just by combining Doppler with contrast-enhanced ultrasound scanning. But we want to get more information. These were just analyses where we draw a region of interest around the whole tumor. And we want to go deeper into tissue characterization. We want to learn about texture. We want to extract all the diagnostic information that is in the image. And here we come to the field of radiomics. There are hardly radiomics papers about ultrasound yet, but I may briefly give you the definition of radiomics, how it is described in Wikipedia. They say that radiomics is a field of medical study that aims to extract large amount of quantitative features from medical images using data characterization algorithms. So that means that we use all the computer power to analyze these images. And for that purpose, here we need to go from a whole region of interest analysis more towards a pattern analysis to a voxel-wise analysis. But the question is, can we easily do that? And Peter Burns already explained to you the destruction replenishment method. And I would like to take that as an example. Yeah. So just briefly, the method works that I inject the microbubbles, then I have a stable level in the blood, then I apply destructive pulses. Yeah. In the area of interest, the new microbubbles will re-enter the tissue, and then I can fit the slope of the curve to get the values about the perfusion. Well, I can do that for such a large region of interest, but in principle, I can also do that on a voxel basis, getting quantitative parameter maps. But the question is, how much will noise here increase when I do so? And will I still get quantitative and reliable data if I do it on a voxel basis? And we did some phantom experiments on that. Yeah, we just took a tube and let the tube be perfused with a defined velocity of liquids, and then we performed destruction or replenishment measurements and analyzed the outcome of the ultrasound measurements in comparison to the given perfusion. And you see very nicely that if we just make a region of interest analysis, we have almost a slope of almost 100% correlation, very good correlation coefficient as well. But you see that if we now go down to a more voxel-wise situation, even in this very standardized phantom situation, we increase significantly the deviation of the fit from the ideal, and we also decrease the degree of correlation that shows that the data becomes less reliable. And just to explain that, what we did here is that we took all the voxels together and took the mean out of all these voxels in comparison to a region of interest that circumscribes the whole area. So that means it's not always easily possible to conclude from a whole region of interest to a voxel-wise information. But on the other hand, you see how much more information is in the voxel-wise image. You see here that you can now suddenly also even see the laminar flow properties in this tube reflected by the higher velocities in the center of the tube. This becomes even more dramatic if we now go into in vivo situations. And here you see the results from 3 different tumors where we measured the perfusion depending on the degree of binning. So we did an automated binning from the whole region of interest to a single voxel analysis, and you see that there's a systematic change in the perfusion values. This is due to the fact that the smaller the voxels become, the more artifacts from motion, from any kind, influence the replenishment kinetics. And we thought, well, this might be an easy way here to define a resolution for such a parameter map that can be used for quantitative image analysis, but just by the way that let the computer automatically bin all the voxels and compare these values. And then you can say, okay, if I have reached a mean value that is not deviating more than 10 or 20% from the whole region of interest, then I'm in a value where I can trust the single voxels. Okay, this is the result, and the nice thing that I can tell you is that if you compare here to the single voxel values where you see almost nothing concerning heterogeneity and differences in the vascularization between these tumors, you can very nicely see that these tumors are differently vascularized if you go to the ideal binning situation, and you see that this tumor here has much more highly vascularized area than this tumor. In the contrary, it also shows you that it doesn't always work, and that in the worst case you may end up in some cases with a tumor where you can only divide it into 3 voxels. And then of course pattern analysis and radiomics analysis will be difficult. But now let's take example where it works. How do we perform these kind of radiomics analysis in tumors? So you can firstly take the B-mode images and extract first order statistics and texture features. Then you can take the contrast enhanced images without functional information, just analysis, for example, number of vessels, relative blood volume, and regional differences in vascularization. Then you can do some statistics on means, standard deviations, and distances between the vessels. Then you can add the functional characterization concerning, for example, blood flow and perfusion. And so you already have 4 groups of parameters that you can extract. And then you can take the best of these parameters, the best distinctive parameters, to form such kind of cluster analysis here to differentiate the tumors. And I would like to briefly explain to you how that works. You may have seen these colored images everywhere, but they are not as difficult to understand as you think. So what you do actually in this very easy way of Euclidean distance approach is that you take the first parameter that you extract from mouse 1, subtract the first parameter of mouse 2, then you take the second parameter of mouse 1, distract it from mouse 2, it takes the square root of that, and as a result you get a value based on which you can rank the different comparisons. And then you can take the mean between 2 and compare the mean with the next ones. So you get an order, an automatic order and clustering. And as you see, if you do that here just with the 3 best discriminating parameters, which were RBV, the image minimum, and the gray value matrix correlation, we can correctly assign all the 3 different tumors to their histological group. Well, I just showed you that this destruction replenishment method has its disadvantages, particularly if we come to single voxel level. And I think there's a really elegant way to replace, at least in some applications, this tool, and this is super resolution ultrasound imaging. And you mentioned already results from Michael Tonté, who started to work on that approach like us around 10 years ago. But there's a big difference in the way he does super resolution ultrasound images and how we do it. So he usually builds scanners that have very, very high frame rates, and he goes for very ultra fast imaging to see the bubbles tracking. We are doing that more with tracking algorithms, and I explain you how it works. We call that method, by the way, motion model ultrasound localization microscopy. So actually we do the same, but with clinical scanners or scanners with a considerably low frame rate. So we have from frame to frame, we see bubbles, or we see voxels that contain bubbles, to be correctly. And then we apply algorithms that are known from radar localization, like the ones that you have in your car, in your navigator system, to calculate the probability of a bubble to have moved to one of the voxels in the subsequent slice. And if you have done that, we can also determine the center of mass within the voxel, and then we connect the center of masses with lines that have almost the thickness of the real size of a microbubble, let's say 2 or 3 micrometers. So we get very thin lines. And by that we get a super resolution image that is clearly superior to the value that we get from the ultrasound scanner. So what can we do with it? First of all, we have then these images with these lines. This is not real lines, it's just drawn by me to illustrate it here. And for example, we can determine the relative blood volume by dividing the area covered by microbubbles to the total area. Then we can measure the distances between the vessels. So how much distance between, so have real some information about the vessel density. We can also make a mean, variance, maximum, medium out of it, which is also difficult if you go for replenishment kinetics. Then we can assess blood flow. And with that method, we can really determine blood flow in individual microvessels, individual capillaries. We can determine the blood flow. And also here, based on that, we can make means, variances, maximum, medians, a lot of parameters. And then we can make mixed parameters. For example, we can determine how are the distances between vessels with high flow, so more the mature ones again, how are the distances between vessels with low flow, and so on and so on. So we applied that approach on 3 different tumor types with different angiogenic properties, and we started with the A431 squamous cell carcinomas, which are highly angiogenic. Then we had the MLS tumors, which is an ovarian cancer, which is a very mixed mature and immature vascularization, and the A449 tumors, which have more mature vessel structures. And you see already here the velocity maps and the direction maps of these tumors look really different. You have to consider that this tumor, by the way, here has a size of about half a centimeter. We talk here about 5 millimeter resolution. 5 millimeter size, so the resolution is in the micrometer range. Okay, but does it really help us to have that high resolution? Can we really use that now to make better diagnosis, to better distinguish the tumors? And so we went back to our radiomics approach. And the first step that we did is that we looked which of the parameters are distinguishing all 3 tumor types significantly. And we defined 5 parameters. Interestingly, these were not perfusion, and they were also not related to blood flow. The most distinctive parameters in our approach were the distances between the vessels. Interesting. We didn't expect it, but that's what came out. And after having these differences between the groups, you then wanted to know how good is the method really to assign, or the parameter to assign each tumor correctly to its histological group. And for that purpose, we used a leaving one out cross correlation method, where we always compare one tumor with all the others and assign it in a matrix. And we found out that for 2 parameters concerning the distances between the vessels, we could 100% assign all the experimental tumors correctly to the histological group. Encouraged by these findings, we thought, okay, now let's go one step further and try that in the clinics. And we had to adapt the tool a little bit to the clinical settings, and we applied it in patients that were treated with neoadjuvant chemotherapy for breast cancer. And the images are not that beautiful as the preclinical ones. I have to admit, I will tell you in a minute why this is the case. But you see that this tumor appears at the beginning considerably hypovascular, which is quite typical for many of the breast cancers. If we then start chemotherapy, we see an early increase in vascularization, which is due to the decrease in tumor cell number and the decompression of the vessels. And then we see in the following the shrinking of the tumors. So we can say from these images, it basically works in the clinics. I have to admit that we still suffer from severe problems that are related to motion. Particularly, the in-plane motion can be quite easily corrected, but the out-of-plane motion really is a big problem for super-resolution ultrasound imaging. And I fully with you, Peter, here that we really need 3D transducers, and they would tremendously help us to bring these technologies more reliable to the clinics. And I would like to go on, after having shown you a lot about functional imaging, to go a little bit more into the molecular ultrasound imaging field. I will not only show you data from these clinically translated BR55 bubbles from BRACCO, but also some data from our own bubbles, highlighting the difficulties you may face with if you're doing molecular imaging. So these are the first bubbles that have ever been translated, the targeted bubbles that have ever been translated to the clinics, which are sulfohexafluoride bubbles with a phospholipid shell here, to which via a polyethylene glycone chain, a targeting unit against the VEGF receptor 2 is linked, and VEGF receptor 2 is one of the main angiogenesis factors. So how do we detect these bubbles here? And imagine you inject a normal microbubble, the accumulation curve in the tissue would look like that. If you inject a targeted bubble, you would have that curve. The information that you would like to have is not the plateau level of the accumulation of the targeted probe. The information you would like to get is this level minus this level, because the bottom level represents the amount of bubbles that are still floating in the blood. Please also note that the initial part of the curve doesn't look much different. So the initial inflow part of the curve is still almost the same, and you will see later data on that for the targeted, non-targeted bubbles. So what we do if we want to see molecular information is that we just apply a destructive pulse after we have known that the targets are saturated by the microbubbles. And then we look for the early replenishment of the bubbles, and then we have a plateau level before and after the pulse that we can subtract to get the information about the molecularly bound microbubbles. And that's how it looks in reality. That's an experimental tumor here before injection of the targeted microbubbles probes. Afterwards, you see a clear accumulation of the VEGF receptor 2 targeted bubbles in the tumors, and after the destructive pulse, most of them are gone. If you quantify that, you see here the plateau level after bubble binding. Here we made the destructive pulse. You see the level afterwards is lower here, so the difference reflects then the molecular binding. We evaluated these bubbles initially for differentiating breast cancer with different aggressiveness, and we tried first animal models, the highly aggressive MDA MB231 tumors and the moderate aggressive MCF7 tumors. And by histological characterization, we could first confirm indeed the more aggressive tumors tend to have more angiogenesis and tend to have higher VEGF receptor 2 level. That's already good. But if you want to move something towards clinical application, you always have to compare it to the state of the art. And if you say that contrast-enhanced ultrasound would be state of the art in this application, we would have to know, are our targeted markers really better in distinguishing the tumors? And we have to say, for these two tumor models, indeed, yeah, we could also see significant differences in vascularization between these tumors. But if you look for the ratios, you see that with the molecularly targeted probes, we have much higher differences. And this is due to the fact that most people think that an angiogenic tumor is always highly vascularized. But this is a complete misbelief. Yeah? An angiogenic tumor is usually hypovascular. If you take the whole tumor, it has only some areas that are highly vascularized because the vessels do not maturate. So what happens is it always produces new vessels, but the vessels don't survive. They immediately get compressed and they die. Yeah? So that's the reason why vascularization often fails as a biomarker of tumor aggressiveness. And if you compare, for example, juvenile fibroadenoma in the breast, it's a highly vascularized tumor, but it's not malignant. Yeah? And the reason is all these vessels, they get matured. And they don't die, they survive, resulting in a highly vascularized tumor. So considering that, you may now say, okay, here, having two mouse models in comparison is really not predictive to a human situation. And we thought that's true. Yeah? And we looked for literature about the data for the expression of VEGF receptor VEGF receptors in breast cancers. And we were really surprised. There are about 300 to 500 preclinical papers about using various types of VEGF receptor 2 targeted probes to characterize breast cancer, but not a single clinical study who really looked at the pathological reality about how is VEGF receptor 2 expressed in inflammation, how it is in benign tumors, how it is in the normal tissue, how it is nothing. Yeah? Imagine that companies spend millions of euros in developing agents without even looking at pathology and having one clue whether their marker can even theoretically work. And so we performed that study here, at least a smaller one in breast cancer. And we compared vascularization that we found by histology with the expression of VEGF receptor 2. And what we could clearly say is, indeed, based on vascularization, we could distinguish normal tissue from carcinoma and normal tissue from fibroadenoma. But the most important differentiation, the differentiation between carcinoma and fibroadenoma based on vascular density in histology was not possible. Yeah? In contrary, if you use VEGF receptor 2 expression here, we could distinguish all the three groups, which is good. And if you now look for the area under the ROC curves, this is also reflected by the fact that if you compare the differentiation between fibroadenoma and carcinoma based on histology, based on vascularization, it's almost not possible, but that you at least get an ROC of about 70 for making it based on the VEGF receptor 2. Whether this is really enough to do a proper differentiation between these tumors and whether this is enough to be implemented into clinics, I'm not sure about it. Yeah, it might be, but further research has to show it. Well, still encouraged by these findings, we applied these bubbles for other purposes and tried whether we can use them, for example, to detect neoplastic conversion of inflammatory liver diseases. And for that purpose, we used a knockout mouse model here that developed liver dysplasia over time and compared the binding of BR55 to the wild-type animals. And we also injected Sonovu to look for differences in vascularization. And in these early pre-neoplastic conversion states, we could not see any increase in vascularization with the functional ultrasound, but we could already clearly depict the increase in VEGF receptor 2 upregulation, which is triggered by macrophages and by the neoplastic cells. So that means we have a clear advantage of using molecular ultrasound here over functional methods. But there are also some problems associated with molecular ultrasound imaging. And this is a quite early study from my lab where we used our own polymeric microbubbles targeted against VEGF receptor 2 and integrins to monitor therapy responses. And here we again used mice with squamous cell carcinomas and treated them with an MMP2 and 9 inhibitor. Yeah? And we started very small tumors that just turned angiogenic. And what you can very nicely see that during the angiogenic switch, we see a clearly increased binding of our targeted microbubbles over the observation time of 8 days. If you now treat the mice with the MMP inhibitors, we see a clear decrease in binding, indicating some kind of therapy response. But what do we really see here? Do we see here an antigenic effect? Who thinks we see an antigenic effect? Ah, nobody, that's good. Yeah? Because it's true. Here we don't see that. Yeah? Because now look at that. Yeah? You don't know if the target decreases. It can be due to a switch in the molecular profile of the vessels, which would be an antigenic effect, or it can just be a cytotoxic effect related to the death of vessels in the tissue. And to figure that out, we then compared the vessel density histologically stained with CD31 and the change of our marker expression. And you see vessel density goes down in a similar way like our markers do. And this clearly shows that these vessels have still the same angiogenesis status, but they are just killed. Yeah? And if you really want to understand the function and the action of therapeutics, you have to puzzle out these two facts. And this information could be easily got if you combine molecular ultrasound imaging with a determination of the relative blood volume. And so I would like to come back to the curve that I showed you how molecular ultrasound imaging works. I told you we get the molecular information from this back part of the curve, but we have here the front part that looks quite similar for the non-targeted and the targeted microbubbles. So our idea was, can't we get the whole information, vascularization, relative blood volume, perfusion, and molecular information, maybe from one scan of molecularly targeted bubbles, if we do it properly with the analysis. And we evaluated that again in mice with breast cancer, and we treated them with an anti-VEGF antibody and compare the outcome also to functional measurements with Sonovu. And you see, first of all, that if we do a first pass analysis with Sonovu and BR55, the results are almost the same. We see a tendency of decreased vascularization under the antigenic therapy and approximately the same values, which confirms that even with BR55 you can reliably determine the relative blood volume and perfusion values. But then you see that if you now go for the molecular information again, and as expected and as I explained already, we have a much higher difference in the responses that is indicated by the targeted bubbles showing their superiority. But now we would have the option also to normalize this data to the data about the relative blood volume, and you would see in this example that we here really have an angiogenic effect and a downregulation of the respective receptors. This is what I wanted to tell you about therapy monitoring and understanding the mechanisms of therapy. Now I would like to really go into the field of ultrasound theranostics. And there were already other movies shown how microbubbles oscillate, and this is another one. And we all agree on the fact that the oscillation of these microbubbles is huge. And by these oscillations, the microbubbles transmit energy, mechanical energy to the vessels and to the cells, and this can be used to manipulate the tissue. And there was one very nice study published 2 years ago by the group of Norway, McCormick has published it, where they used just contrast enhanced ultrasound in an interoperative setting to permeabilize the pancreatic tissue to improve neoadjuvant chemotherapy outcome. It was not really a prospective study. They compared their outcome to a historical cohort, so there's still some uncertainty in it. But the results were encouraging, and they could show a significantly prolonged survival of the patients that were treated with Sonoporation in comparison to the historical group. So that means Sonoporation may be really an enabler of better chemotherapy response in patients. And as it was already mentioned, the group in Toronto around Huninen here, they are very intensely exploring Sonoporation in the field of the brain, where they try to open the blood-brain barrier with microbubbles, enabling better chemotherapy delivery here. And I think the early clinical trials on that are encouraging. Another group in France around Carpentier, they developed an implantable ultrasound device also for patients with brain tumors. So the ultrasound device stays in the body, it's in the skull here. And even always if they give chemotherapy, because these tumors are usually not curative, they can apply additionally focused ultrasound and contrast enhanced ultrasound to improve chemotherapy accumulation. We also already worked quite a bit on the field of bubble-mediated drug delivery, and for that purpose we usually use a microbubble platform that consists of N-butyl cyanoacrylate. So more or less we produce the bubbles by adding super glue here to a buffer solution. Then we rapidly stir the solution. By the stirring we have inclusion of gas, and then the polymerization takes part around the gas, forming a microbubble. And that's how such microbubbles look like. It's not the nicest TEM image, but at least it shows you. Here are some microbubbles where we had some dyes in it here. And if you then apply high energy ultrasound pulses, we can destroy these microbubbles. And as you can nicely see, they fall apart in multiple small fragments of polymers of about 200 nanometers length here, and then release their content and also transmit the mechanical energy. So it's important to understand that these bubbles are completely different than, for example, phospholipid bubbles, which have just a very flexible and thin layer. And from first 10 images, we thought that the shell of these bubbles is only around 50 nanometers thick. But this was more a staining artifact. If you really look to these microbubbles under a STAT microscope, yeah, or confocal microscopy, you see that the shell thickness of these bubbles is much, much thicker. So we have here a bubble with a size of about 2.2 micrometers and a shell thickness of 300 to 400 nanometers. And you have to imagine that these bubbles look like more like a web. Yeah, so these polymer fibers are interwebbed, somehow cross-linked. Yeah, they form a really like a sponge. Yeah, and this of course gives us great options to load something into the sponge. You consider here we have 400 micrometers. That's a lot to load something in. Yeah, and we can easily load, for example, nanoparticles, drugs, and dyes into this microbubble platform. We evaluated these bubbles. We also wanted to apply these bubbles to open the blood-brain barrier. And we were very successful with that. We tried that in mice, yeah, and all the mice died. And we looked why they died, and they all died by brain edema because we opened the blood-brain barrier too much. So intervention was successful, but we did too much. So we thought we need to have some kind of control, how much we open the blood-brain barrier. And so our idea was that we take small iron oxide nanoparticles, like they are used for MRI imaging, embed them into this sponge-like shell of the microbubbles, and then we let the bubbles burst. And during the bursting, we have a release of the nanoparticles from the microbubble shell, and then we can take simultaneously MRI to monitor the accumulation, the opening of the blood-brain barrier. In addition to that, we injected fluorescent dextran of 70 kilodalton as a model drug to confirm whether we could really transport drugs over the blood-brain barrier using this approach. And here you see that the method really works. If we only inject the microbubbles, nothing happens on MRI. If we only perform ultrasound, nothing happens. Yeah? But if we apply ultrasound and inject the microbubbles, depending on the time of scanning, we see an increasing MR signal related to the accumulation of these iron oxide nanoparticles. And we also see a quite strong accumulation of our fluorescent probes outside the vasculature in the brain tissue. Even more, we could show that by varying the intonation time and also the microbubble dose, we could also change the depth of drug penetration into the brain tissue. We measured here the distance of drug signals from the respective vessels, and we saw that we could trigger that to a few hundred micrometers, which is really important if you want to use it for therapy. And not only think about treating tumors here, we also need to think about bringing growth factors, for example, to the brain in the field of neurodegenerative disorders. In the next step, we wanted to know how big are these gaps. Can we even transport larger molecules over the blood-brain barrier? We tried that with polymers of a size of about 12 nanometers. This is the size of an IgG antibody that is, for example, used in immunotherapy. And we could very nicely show that also for these big molecules, the sonoporation of the brain tissue works, and you can nicely transport the drugs into the brain tissue. And here you see a result of a fluorescent model drug here. If we do not apply sonoporation, and if we apply sonoporation here, I think the difference speaks for itself. In the next step, we applied that on peripheral tumors, and something that you should keep in mind is that the effect in peripheral tumors is completely different than the effect in the brain. In the brain, you really have an opening of the tight junctions of the endothelial cells. In peripheral tumors, there are no tight junctions between the endothelial cells. They may rather have gap junctions or may have a fascia adherentes, so very loose contacts. And in peripheral tumors, the gaps in the vasculature have a size of around 100 to 300 nanometers. So that means that the vascularization is completely very leaky. And so the effect that you have here from sonoporation may not come from vessel permeabilization directly, but still we could show, and we applied it here with liposomes, which may still be a little bit too big, that also in peripheral tumors you could significantly enhance the accumulation of molecules if we perform sonoporation. I will come later to that, why it is so. In addition to the improved accumulation, and this might be even more important, particularly if you talk about stromal tumors and desmoplastic tumors like pancreatic cancers or prostate cancers, we could significantly improve the penetration of the drugs into the tissue, so we could help to overcome the stromal barrier. And you already mentioned here that most probably the stromal untightening here is a very important factor for successful tumor response. It's in my opinion more or less the most important one, because the leakiness of vessels is usually high. But even if you look into the nanomedicine field, they show high accumulation of nanomedicines, but no good response. And the reason is these materials, they just leak out of the vessels and then they just are deposited outside the vessels, but then the stromal barrier comes and they are not transported to the tumor cells. And I think here ultrasound can play a big role, and we even show that here with intravascular regular microbubbles. Already here we have a quite strong untightening effect on the stroma. You can even go one step further and then say, okay, if this works, then we can also load drugs directly into the microbubbles, and make the bubbles targeted, and then direct the bubbles to the vasculature and let them explode. Then it's even closer to the vascular wall. The effect then on the stroma and on the accumulation might be even bigger. And we tried that as well. So we took the loaded microbubbles, we injected them in mice with tumors let them bind then performed destructive pulses and looked whether our fluorescent model drugs accumulate in the tumors. And indeed, we could show that if we don't perform ultrasound, we have after a certain time a washout of these bubbles, no drug accumulation, but that we can even with few pulses release the drugs into the tumor tissue. There's always a debate for these drug delivery processes, whether you should do a co-formulation approach or whether you should inject the bubbles separate from the drugs. And this can have quite different results. And I show you here an example where we didn't take a drug, but genes, because we wanted to transfect the tumors. And we used lipoplexes encoding for DS-RET mRNA. That means that we have a fluorescent protein that will be expressed by the cells once they get transfected. And we either bound the lipoplexes to the microbubbles and injected it as one construct, or we applied it separately and first injected the lipoplexes and then injected the microbubbles and did our Sonoporation approach. And as you will see, the results are quite different. If you do a co-formulation, we mostly transfect the endothelial cells, but not much of the cells behind it, most probably because we have a direct shooting of these lipoplexes into the permeabilized vasculature. However, if we first inject the lipoplexes, they will accumulate in the tissue via the enhanced permeability and retention effect. And then we apply ultrasound destruction. Then you see that we then start to transfect the cells behind the vascular wall. And so we have to think about what do we really want to transfect, where do we want to transport our drugs to. So we have options to vary here. If you want to be more on the vascular side, you may do rather a co-loading approach. If you want to attract the cells behind, you may rather inject it separately. Of course, it also depends on the drug and the molecule here and the gene and so on and so on. Encouraged by these preclinical findings, 1 year ago, a little bit more, we also started a clinical trial on Sonoporation of patients suffering from triple-negative breast cancer. Here we used regular phospholipid microbubbles as a contrast agent, and we applied a destructive pulse to improve the accumulation of chemotherapy by increasing the perfusion of the tumors by the Sonoporation approach. And this is just an image that shows you here how perfusion changes if we perform Sonoporation during chemotherapy infusion. You see from this almost hypovascular tumor over 20 minutes, we see quite some decent vasculature after the Sonoporation procedure. And I think that is the major mode of action of Sonoporation in peripheral tumors. It's not permeabilizing the vasculature, it's opening vascular shunts because the vessels are compressed. And as you mentioned, we untighten the stroma, so we reduce interstitial pressure, and by that we open the vasculature, and then we just have more sites that really supply the tumor with a chemotherapeutic. So it's more a vascular untightening effect than a permeability increasing effect. And this is, I think, the big difference between peripheral tumors and the brain. With these words, I think I'm already at the end of my talk. I think I more or less saved your lunch break here, and I would like to conclude here. I think ultrasound has a great potential as diagnostic imaging modality. I showed you that radiomics analysis are possible with ultrasound, but we still need to improve its quantitativeness to reduce the user dependence. And I think a big enabler will be going to 3D acquisition. I introduced Superresolution as a novel method to perform vascular characterization. I think it has great potential, and I know several companies are already thinking about implementing it into their clinical devices. I showed you that targeted microbubbles have a certain value, particularly for therapy response evaluation, that they can be superior and faster with respect to the detection of drug responses in comparison to the non-targeted microbubbles. I think there is a really big chance for using microbubbles for therapy. So we already know that Sonopermeabilization, how I call it now, that it works in the brain, and we also can now show, and other groups also show similar results, that it exquisitely also works in peripheral tumors, so we should go for that. Yeah, I think there's also on the long run a big chance for using microbubbles for direct drug and gene delivery, although your argument was right that the costs are here certainly one of the main limiting factors here that need to be considered. Nevertheless, for many ultrasound applications, interesting clinical translation is ongoing, and if you really understand the mechanisms of action of these bubbles, but also side effects, I think it will be a great tool. With these words, I would like to end my talk. I thank all the people from my lab, my collaborators, the funding agencies, and you for your attention, and hope that there will be at least a few questions.